Welcome to the second video for the cellular energy unit. In this video, we will talk about the energy conversions that occur during cellular metabolism. In order to be alive, all cells must have a source of energy that they can harvest from their environment and a source of carbon in order to build their various macromolecules, such as proteins and phospholipids. Organisms being classified based on these energy and carbon sources. All organisms obtain energy in one of two ways, either from the sun, in which they are called phototrophs, or from the breakdown of chemical compounds called chemotrophs. All organisms can also obtain carbon in one of two ways. They can obtain it from inorganic sources, such as carbon dioxide. These organisms are called autotrophs. Or they can obtain it from organic molecules, such as carbohydrates. These are called heterotrophs. Organisms are then classified based on the combination of these terms. For example, plants obtain energy from the sun and carbon from carbon dioxide, so they are classified as photoautotrophs. Various bacteria are classified as photoheterotrophs, while other bacteria are chemoautotrophs. Humans, such as yourself, would be classified as a chemoheterotroph. Cells require both an energy and a carbon source so that they can perform all the chemical reactions necessary to support life. The sum of all chemical reactions inside a cell is defined as the cell's metabolism. There are two branches of metabolism. One takes macromolecules, such as carbohydrates or fats, and breaks them down into smaller subunits, such as sugars or fatty acids. The breakdown of molecules is called catabolism. The opposite metabolic branch involves the building of macromolecules from the individual building blocks. This is called anabolism. You can visualize these branches during digestion. You take in macromolecules in the food you eat and break them down into building blocks to be absorbed into the body. Your body then takes those building blocks, such as the amino acids broken down from a steak you just consumed, and uses them to build cellular proteins that are acquired through translation. Catabolic reactions release potential energy that has been stored in the covalent bonds of the macromolecules, which is captured by the cell as a molecule of ATP, the main energy currency of the cell. This ATP is then used to provide energy required for anabolic processes to occur. This is similar to you working to earn money and putting it into a savings account, and then withdrawing it to pay for things you want later. A molecule of ATP is equivalent to your savings account. It is a stored form of energy that a cell can access later when energy is required to do work. ATP is composed of adenine, the 5-carbon sugar ribose, and 3 phosphate groups. You should recognize this as a ribonucleotide triphosphate. It is the same nucleotide that would act as an adenine building block in an RNA molecule. The 3 phosphate groups of ATP are where the majority of potential energy is stored in this molecule. Remember that like charges repel, so it takes a lot of energy to hold these 3 negatively charged phosphates together. The most common use of ATP is to break the high energy bond holding the 3rd phosphate to the chain, which forms a molecule of ADP. Sometimes a pyrophosphate or 2 phosphates are released instead of just 1. In this case, we form a molecule of AMP. ATP hydrolysis to ADP involves the use of water to break the bond holding the 3rd phosphate of ATP as depicted in this figure. This is an exergonic reaction and has a delta G of negative 7.3 kcals per mole under standard laboratory conditions. The negative 7.3 kcals per mole of energy that is produced from the exergonic hydrolysis of ATP can be used in energetic coupling reactions to power other endergonic reactions in the cell. This is what occurs during anabolic processes. Remember, for coupling to be successful, the overall delta G for the combined reactions must be exergonic, and a product from reaction 1 must be a reactant of reaction 2. In this case, the shared molecule is the inorganic phosphate, and the overall delta G is a release of 4 kcal per mole of energy. ATP hydrolysis actually has an intermediate delta G when compared to other phosphorylated molecules in the cell. This is important for cell biology because the reverse reaction of ATP hydrolysis, ATP production from ADP and inorganic phosphate, would have a positive delta G and need 7.3 kcal per mole of energy put into the system in order to be formed. Reactions with a higher negative delta G will produce enough energy to power the production of ATP, which means during catabolic reactions, in this example, that means that the phosphoenol pyruvate reaction could be coupled to ATP production because 14.8 kcal of energy would be enough to provide the required 7.3 kcal of energy, leaving a negative 7.5 kcal overall delta G for the coupled reactions.